Okay, hello everybody. How are you today? Any comments? Any worries? Can you hear me? Yes? Is someone alive out there? Good. Thank you. No comments. Yeah. If you the exam is a lot of lectures, but some of them have very few pages. Like the first one only has one page. And it's all kind of a sequence. You build on the knowledge, and it's fun, I think. Um, So, the final is worth 18%, right? The other two exams are worth 16% each. So it's going to be okay. I think if you study all along, you're going to do very well. There's many students that don't come to the lectures. Right now, we have, how many? 61. No, it's their loss because they can listen to the recording. But why wouldn't they be here? This is the time that they signed up for the lecture. I never understood. Anyway, let's see. Let's start. Okay, so what we're going to do today, we're going to continue with the different types of neurotransmitters. And going to talk about circuits, because everything up to now has been very simple, like from one presynaptic neuron to another presynaptic neuron. But that doesn't exist in our central nervous system or peripheral nervous system. The neurons are all interconnected with each other, and there's a lot of circuitry going on. So today we're going to get into those circuits, types of circuits that the neurons establish. And if we have time, we'll talk a little bit about the spinal cord. Okay, so today we're going to talk about neuropeptides as neurotransmitters. So what are neuropeptides? Peptides, remember, they're made up of amino acids. Therefore, those peptides have to be synthesized. And they have to go through transcription, translation, and then go from DNA to messenger RNA, then to go to the ribosome, where the proteins will be synthesized. So the synthesis of these neuropeptides occurs in the cell bodies of the cell. So they're synthesized in the cell body, and then they're packaged, because they have to be packaged in synaptic vesicles, right? And then those synaptic vesicles are transported down the axon to the axon terminal. Some of these neuropeptides can be excitatory or inhibitory. It depends on what type of receptors are in the postsynaptic membrane. If they open chloride channels, then it will be inhibitory because it becomes more negative. It goes from the resting membrane potential, which is minus 70, to becomes hyperpolarized, can go to minus 80. And therefore, it's much harder to depolarize. Or they can be excitatory. If they open sodium channels, then sodium rushes in, and the resting membrane potential now goes up, and it becomes depolarized. Okay, so one of these neuropeptides is known as substance P, and it is involved, it's a neurotransmitter that is involved in the perception of pain. So the pain sites in the brain are located in the cerebral cortex, as you can see here. So you see the sensory neuron, which would be at the end, would have a receptor for pain. Pain receptors are very simple receptors. So the eye is a very complex receptor for photons. But pain receptors are very simple. So when they are activated and the action potential is established, it travels down to the spinal cord, and then there it can synapse with another neuron that brings the action potential to the thalamus, and then from the thalamus it goes to a specific area in the brain. That's where, and this will allow the perception of pain. So the action potential has to travel, has to be strong enough to travel up to the brain. So fortunately, we have some neurotransmitters that decrease or mitigate the sensation of pain. So they're pain relievers. And how can they do that? So they activate GABA receptors. So you know that GABA is the inhibitory neurotransmitter in the brain. So if GABA activates its receptor, it can be a chloride channel or it can be a potassium channel. Then the potential at the membrane becomes more negative, becomes hyperpolarized, and that's why it is inhibitory. So the enkephalins, they act at GABA receptors. They influence GABA receptors. And they can be considered, they're opioids, and they are 200 times stronger than morphine. So that's amazing. And actually, it's thought that the acupuncture, which relieves pain, will activate the release of enkephalins and other opioids. So you have endorphins and dynorphins and enkephalins. All these are analgesics, our own analgesics. When does your body release this? When you're injured. Not only when you're injured, for example, if you are doing a lot of exercise, like a marathon runner that runs for, well, the most efficient marathon runners can do the 26.5 miles in 2 hours, more or less. That's amazing. But when they finish, it's a lot of effort, wear and tear on the muscles, and that could lead to enormous amount of pain. So this is a time where our own opioids will be released to decrease the pain. So these marathons describe their feelings as being high, almost like having taken opioids. So it could be injured, it could be, all kinds of stimuli that lead to pain. Okay, so here are the neuropeptides.
Do enkephalins give you pain or relieve pain?
No, they relieve. They're analgesics. The pain is the substance P, right? That's the neurotransmitter for pain. Enkephalins and endorphins and dynorphins, they are analgesics. They are pain relievers. Okay? Okay, then we have gases. I mean, a gas being a neurotransmitter. So carbon monoxide seems to be a neurotransmitter, but there's not much known about this. It's very controversial. But nitric oxide, NO, is definitely a neurotransmitter. And it's interesting because nitric oxide, if it's released in high concentrations, can be very toxic. So one of the mechanisms that white blood cells, such as macrophages, can cause the death of cells that are infected by microorganisms, viruses, or so on, is they release nitric oxide. And nitric oxide can be very toxic if it's released in high concentrations. You see here toxic in large quantities. So it's a strategy that is used by these white blood cells, the macrophages, to kill the cells that are infected. But when it's released in small amounts, it's beneficial. So what does it do? It causes vasodilation. So it acts on smooth muscle. So this enzyme nitric oxide synthase, there's three different types of nitric oxide synthases. One present in the neurons, another present in macrophages, another present in the endothelial cells. And these enzymes act on arginine. So arginine is an amino acid. So I think you're getting the idea that a lot of these neurotransmitters have amino acids as precursors. It's a very efficient way. So we have our own precursors to make these neurotransmitters. So for nitric oxide, the precursor is arginine, and this enzyme, nitric oxide synthase, so the name synthase, it synthesizes nitric oxide from arginine. And nitric oxide can cross freely the plasma membrane, so it's lipid soluble, so it has no problem getting into the cell. So it acts on smooth muscle and causes vasodilation. So it causes the smooth muscles to relax. And what does it mean, vasodilation? That means that the blood vessels the diameter of the blood vessel increases. So the question is, why is carbon monoxide controversial? Because there's not enough studies. So, sometimes the conclusions that come from studies done in labs do not always agree with each other. It depends on who is doing the experiments. It depends on temperature, on so many different factors. So carbon monoxide still has the knowledge on carbon monoxide as a neurotransmitter still needs to be further investigated. Oh, how much carbon monoxide is considered toxic? Okay, I don't know exactly in volume, but you know that if you're stuck in the car and the car is working, the motor is on, it releases carbon monoxide and the person can die, right? So it's interesting. You're interested in the carbon monoxide, but there's very little knowledge about. But I know it's an interesting kind of statement that a toxic substance can be used as a neurotransmitter. The same for nitric oxide. If there's too much, it's toxic, like the one released by macrophages. Okay, so this nitric oxide can be used for treatment for disease conditions. For example, nitroglycerin is a substance that can, if it's metabolized, it produces nitric oxide. Is there a reason why carbon monoxide is very undetectable? Well, we do have now sensors for carbon monoxide. So it depends on its diffusion rate and so many different factors, right? I'm not very well versed on carbon monoxide, I must tell you. Okay, so we go to the nitric oxide. So nitroglycerin. So, if someone is having like a heart attack or something, you can take nitroglycerin, you give the patient nitroglycerin. Why? Because nitroglycerin can be metabolized and it produces nitric oxide. What does nitric oxide do? It increases the diameter of the blood vessels. Therefore, there's more oxygen being pumped into the heart. There's more oxygen being pumped to the heart, and that makes the muscles, the cardiomyocytes, work better, right? They have more oxygen. So this is one of an advantage of this nitric oxide. Also, it's used for Viagra because it causes vasodilation. You can see here. Here's the application to the heart. So nitroglycerin and Viagra, what they do, they increase the levels or they increase the action of nitric oxide. So nitroglycerin, when it's metabolized, it increases the amount of nitric oxide being produced. Oh, okay. The Viagra uses a different mechanism. It prolongs the life of nitric oxide, or the downstream, the cascade, the downstream events that are initiated by nitric oxide. But the final effect is the same, is to increase the diameter of the blood vessels. Okay, Professor, to clarify, all these neurotransmitters we went over, such as acetylcholine, amino acids, biogenic amines, etc., are found in different parts of the body, correct? Like either CNS or PNS. Yes, but for example, acetylcholine and noradrenaline, they're found in the PNS and CNS. So, it all depends on this network of neurons that they are cross-talking with each other, and they form these pathways that are very specific for the effect that they are going to have, the ultimate effect that they're going to have. And when we study the brain, so we're going to finish this, which is sort of the nervous system in general, to have an idea. And then we're going to focus on the central nervous system, so the spinal cord and the brain. And we will go over where these different neurotransmitters are going to be released and what is their function and their importance and relevance. But yes. Okay, so here we have the effect of this gas. There was the discovery of nitric oxide being a neurotransmitter led to a Nobel Prize win, because it was so unbelievably, was hard to believe, right? That a gas could be a neurotransmitter. And think about it, its effect is very quick because it's a gas and it will diffuse out of the sight very quickly. You have to design the experiments that had to be designed to show that nitric oxide is a true neurotransmitter. Okay, so we have all these neurotransmitters, and they cannot be around for a long time. They all have their what we call half-life. So they are released at the axon terminals, they diffuse across the synaptic cleft, they bind to their receptors, and then they're there for a limited amount of time. So how do you get rid of these neurotransmitters? Well, some of them. Okay, so there's a question here. The doctors used to give NO to pregnant women in labor. Well, it could be because because its effect to cause vasodilation and to improve smooth muscle activity, because if you have more oxygen arriving at the smooth muscle, then it will work better. The same principle with the nitroglycerin, right? So there's more oxygen arriving at the heart muscle, at the cardiomyocytes, so they will work better. You provide more oxygen, you can make more ATP. That's the whole idea. And the same for pregnant women. Interesting. You guys have very good comments. I like this class a lot. Okay, so yes, so why? So, actually, there's a train of thought. Some laboratories have the idea that giving Viagra to Alzheimer's patients will be beneficial. Why? The same principle, vasodilation, more oxygen. So maybe that will improve brain function because remember, brain is very active. It needs a lot of oxygen, a lot of ATP. But think about it. All kinds, you're so desperate to find a way of improving and stopping the progression of Alzheimer's that, you're very creative. Think about all these different mechanisms that can be altered to prevent or stop the progression of Alzheimer's. Okay, so how do we get rid of the neurotransmitters? By diffusion. So they are released at the synaptic cleft, they bind to the receptors in the postsynaptic membrane, and then they come off. They're not permanently attached. They come on and off. So then they will diffuse away from the receptors. So that's one way. And then they can be taken up by astrocytes. So here, this is a synaptic axon knob, synaptic knob for glutamate. Remember what glutamate is. It's an excitatory amino acid in the CNS, in the central nervous system. So you have the action potential coming down. At the axon terminal, glutamate is released. It diffuses across the synaptic cleft. It binds to the glutamate receptors on the postsynaptic neuron. So this would be, let's say, a dendrite, for example, of the postsynaptic neuron. And now what happens to the glutamate? Some of it is going to diffuse. So the green area here represents an astrocyte. Remember, that's a type of glial cell. And the astrocytes are in very close proximity with the neurons. They can give neurons lactic acid so that they make more ATP, help them make more ATP. They make a connection between blood vessels and neurons and so on. But another important function is they have transporters so that the glutamate diffuses and gets into the astrocytes and then where it's going to be metabolized. So one way of getting rid of the neurotransmitter is they diffuse, and sometimes they can be taken up by astrocytes in the proximity. Then you have some enzymes that can degrade the neurotransmitters. So you remember acetylcholinesterase, which is located right at the synaptic cleft. So acetylcholine would bind to the receptors and they would come off. And you have acetylcholinesterase right there that will metabolize the acetylcholine. Part of it is going to be, it's going to be used, it's going to go back into the presynaptic knob and it's used to synthesize more acetylcholine. So question here, isn't the problem with Alzheimer's that the neurons are like fading away? Yes, the neurons are degenerating, what we call fading away. A more scientific way of saying fading away is degenerating. And the synapses degenerate. So the communication between one neuron and another degenerate. So how can Viagra help with that? So what it means is that those that are surviving, that have not degenerating, by offering more oxygen due to vasodilation, you can improve their function. So we're trying to make the neurons that are surviving and that are not degenerating. Let's help them be more efficient, more active. Okay, exactly. Exactly, Sophia. You got it. It's more prevention. Prevention, yes, but also to allow the neurons that survive to become more efficient. So remember, it's the same myasthenia gravis. Remember that disease that is caused by the degeneration of the acetylcholine receptors. So it's an autoimmune disease. So you give acetylcholinesterase inhibitors so that there's more acetylcholine in the system. So the few receptors that survive, that are not destroyed by our own white blood cells, you make them more, you have more acetylcholine binding to them. In this case, in case of Viagra, you are providing more oxygen to the brain. Doctors prescribe Viagra to patients with Alzheimer's. Like, how common is it to give? So this is all, um, it's all in preclinical stages. So there's so many different strategies, and the patients, they are so well. The patients that have and their families, let's say, are so eager to find something. So Viagra can be prescribed. It doesn't mean that it will help all patients, right? So we're having more and more personalized medicine. What does that mean? Even to treat cancer, right? One form of cancer cells is going to be different of the cancer cells in another patient. So if we can do biopsies and determine what type of cancer cells, what is the mechanism that led to those cells become cancer, then one can devise a treatment that is more precise. That's precision medicine. That's really the aim of nowadays, is to gather more and more and more information to be able to treat each patient on its own. So, for example, in Alzheimer's, 2/3 of the patients are females. So, and there's very few studies done with females because it's hard to have a model like the rodents. They have extra cycle. They don't have menopause, but they go to extra cycle. So it's very hard to model Alzheimer's in a female model. So all these are, a lot of very interesting issues that we are all trying to address. Okay, so the reuptake into cells. So, we talked about the neurotransmitters, the transporters for dopamine, for norepinephrine, and so on. So that here you see a glutamate transporter that brings glutamate into the astrocyte. But you could have other types of transporters on the presynaptic neuron, and this would bring dopamine inside, or norepinephrine and serotonin also can be taken up by their specific transporters. So all these are proteins that are specific for these neurotransmitters. And then they come into the presynaptic knob, and right there you have enzymes that can destroy, like monoamine oxidase and COMT. Those destroy the norepinephrine and dopamine. So several ways of getting rid of those neurotransmitters. They have to be, their presence and their levels have to be highly, highly regulated. Now, don't you think that this is also fascinating, how it all evolved, how all this happens, and we don't have to think about it. And when we are healthy, all this happens automatically, autonomically. So Don't some state to need less activation energy for next contraction? I don't quite understand what you are saying, Sophia. If you can write it again. Okay, so now we have action potentials traveling down the axon, the neurotransmitters being released in a very specific manner. But how can this release and this conduction of the action potential be optimized? So can the action potentials, the conduction of the action potentials from one neuron to another, can it be regulated? Can it be altered? The answer is yes. And here you have, I'm going to give you the example of 6 different strategies in which the conduction of the action potential can be altered. So remember, I gave you this notion that the action potential is an all or none response. So once you reach that threshold of minus 55 millivolts, then the spike of depolarization goes up. But how can this action potential be along the axon? How can it be altered? So these are all chemical ways of altering the conduction of the action potential. So some chemicals can block the channels, the sodium channels. We talked already about Novocaine. Well, there's these chemicals that are present in the pufferfish. So pufferfish is a delicacy that is eaten in Japanese restaurants. And if you ever order this, make sure that the gland that produces tetrodotoxin is removed, because these two chemicals, tetrodotoxin and this STX, they block the voltage gated sodium channels, so they can cause paralysis. And this is a way of the pufferfish surviving, right? They release this toxin and the prey will paralyzed. Okay, how about increasing the threshold or decreasing the threshold? So what is this? What do I mean by increasing threshold and decreasing threshold? So threshold is minus 55 millivolts. So if you increase the threshold, what does that mean? That means that you cause hyperpolarization. So instead of going from the resting membrane potential that is minus 70, going up to minus 55, now if you increase the negativity, if you hyperpolarize the membrane, so it goes from minus 70 to minus 80, then you're increasing the voltage required to reach threshold. Right. Right, you need more sodium. So you're increasing the threshold, you're increasing the amount of voltage that needs to be generated to reaching threshold. You need to increase the amount of sodium, exactly. So that's the principle of anesthetics and hypnotics, tranquilizers and anesthetics. They increase the threshold. So the membrane potential goes down, becomes more negative. So how about to stimulate? Think about caffeine, right? So if we drink a lot of caffeine, keeps us awake. So that means that we are decreasing the threshold. That means that the stimulus that is applied to the neurons, the strength has to be much lower than if we don't drink caffeine. So what happens? The threshold goes down. What does that mean? That means that the sodium channels will be easier to open, the voltage gated sodium channels. So instead of the membrane potential, instead of being minus 70, then it might go to minus 60, or you increase the ability of the sodium channels to open. So what about if caffeine doesn't have any effect on you? Well, that's true. Because the sensitivity also depends on the metabolism. So there are enzymes known as P450 enzymes that we have that metabolize chemicals, right? That's a way of getting rid of toxins. It's to metabolize them. So caffeine can be metabolized by these enzymes. So if there are different forms, different isoforms of these enzymes, and those people that are less sensitive to caffeine, one of the factors can be that their P450 enzymes are more efficient at metabolizing caffeine. So that means that you will need much, much more caffeine to have the same effect than in a person that has less efficient enzymes that metabolize caffeine. So that's one factor. Okay, so they help the receptors, so they can be already partially depolarized. So that's the idea. So to decrease threshold, that means that the membrane is already partially depolarized. So it's going towards the threshold. So this is how caffeine, nicotine, and these chemicals can work, the strategy. How about now, well, chemicals like botulin or Botox, remember what the effect is? They prevent the synaptic vesicles from fusing with the plasma membrane, and therefore there's no neurotransmitter being released. So blockers of neurotransmitter release, another strategy. Another strategy is to block the receptors. So there's this chemical curare that was used by indigenous people as at the heads of arrowheads. So when they released, the arrowhead would release this chemical and would paralyze the prey. So that's another way of altering the conduction of the action potential. So after all, what you're trying to do is alter the opening and closing of the voltage gated ion channels. And then you have inhibitors of the enzymes that metabolize the neurotransmitters. So the anti-cholinesterase inhibitors. Okay, so although the conduction of the action potential down the axon is an all or none response, it can be modified by temperature, myelin, by the diameter of the axon, and also by all kinds of chemicals, which gives us a way, a strategy to treat conditions where you need to alter the conduction of the action potential. So all this knowledge is very important, and it takes a lot of effort to come up with these results and this knowledge. So I hope that you give them enough recognition. Okay, so we finished the neurotransmitter chapter. Now let's focus on the postsynaptic neuron. So the postsynaptic neuron is the integrator. So it can be overwhelmed by all the synapses formed with the presynaptic neurons. So remember that there are 3 different types of synapses that can be formed. So we have axodendritic. So here's the postsynaptic neuron. Here's one dendrite to make things simple. And you see that this axon terminal synapses with the dendrite. So the neurotransmitter is going to bind to the receptors right on the dendrite. So this is an axodendritic synapse. If the dendrite, if the axon terminal synapses with the cell membrane of the postsynaptic neuron, then this is axosomatic. Somatic means cell body. And then you have axoaxonic, where the presynaptic axon synapses with another axon right here. So at the axon terminal. And here's a modulator. In this case, what is happening is you're modulating the conduction of the action potential down the axon. And you will understand this. I have a few more slides to explain this action here of the axoaxonic synapse. Okay, this is what is shown here. So here's the postsynaptic neuron, and now you have two presynaptic neurons. But look here, the blue neuron is synapsing at the axon of the yellow presynaptic neuron. So you have here a communication occurring at the axon terminal. So this means that this neuron can either generate inhibition or facilitation, or it can facilitate the propagation of the action potential from this yellow neuron to the purple neuron, or it can inhibit the propagation, can stop it. So how can that happen? So you see here the blue and the yellow forming an axoaxonic synapse. So suppose that this neuron has the neurotransmitter GABA, is a GABAergic neuron. So it's going to release GABA. So GABA is an inhibitory. It opens up chloride channels. So the chloride is negatively charged. It will go in and will make this more negative. So when the action potential is traveling down here, it has to generate, to open the sodium gated voltage channels, it's going to be much harder, because instead of having the resting membrane potential here being -70, it might be -80. And therefore, the signal has to be much stronger. The action potential is going to be slowed down. So this is the idea that the GABA is the brake in the car. So by increasing the negativity at the axon terminal, it slows down the transmission of the action potential, or from the propagation of the action potential from one neuron to the other. So it makes it more negative. So that's here, it's producing inhibition. It's counteracting the effect of the action potential traveling down the yellow neuron. What happens now if this blue axon, or its blue neuron, has the neurotransmitter serotonin? So what serotonin does, it opens sodium channels. So sodium will come in, so it will make more positive, closer to threshold. So this will make the action potential more efficient. So it facilitates. So the axoaxonic synapse is a modulator, because it modulates the transmission of the action potential from one neuron to the other at the axon site. What would be the purpose of inhibiting the action potential? Well, it's very, very important. Look, because you don't want all the neurons to be active at the same time. And for example, in Parkinson's disease, the dopaminergic neurons in this case are inhibitory. And if they die, then the person has tremors. They cannot inhibit the action potential. They cannot inhibit movement at the muscle. So you do need, so if not, you also go to epilepsy. Think about epilepsy, which is all these action potentials occurring at the same time because you inhibit GABA. You need to prolong the effect of GABA to put a brake. You don't want just accelerator. Think about the car. When you're driving down the road, you cannot just use the accelerator to control the movement of the car. You need a brake to stop it or slow down. So it's exactly the same what happens in our brain, in our nervous system. We do need a way of slowing down the action potential. Okay, so what happens at the postsynaptic neuron? What decides if the action potential is going to travel down or the change in potential, the impulse is going to be transmitted from the pre to the postsynaptic neuron. So that's a process known as summation. So you're adding up the effect of all these presynaptic neurons. So the integrated, the postsynaptic neuron, the effect is the result of the summation of each of the effects of each of these presynaptic neurons. So the summation you can have spatial or temporal. So summation is the result of the action of all the presynaptic endpoints. So it can be spatial or it can be temporal. So what does this mean? So here you have a presynaptic neuron and the axon terminal making a synapse with a postsynaptic. Okay, so what happens here? Focus here. So you see here many different axon terminals. Now, envision a scenario where this axon terminal will repetitively release neurotransmitter. So it's the same axon terminal, but you have the stimulus is going to be very frequent. So it's like a jackhammer. You're going to zzz, right? So each time you're going to release a little bit of neurotransmitter. So adding up all of it, it might reach a level that is threshold. So that means that's what the summation is. It's temporal because it's the result of the same presynaptic neuron releasing and be activating the postsynaptic neuron continuously. So then the effect, the summation, the result of this frequent stimulation can change the potential to reach threshold. How about spatial? Now, spatial, instead of having one presynaptic neuron stimulating it continuously or frequently, now you have several neurons, several axon terminals releasing some neurotransmitter and causing a certain level of depolarization. And then adding all that up, it might reach threshold. So you can have the summation, the result of the effect of these presynaptic neurons can be temporal or spatial. So you see here another view of this. So here's one, this is really simplified. It never happens that you have one axon terminal synapsing at a postsynaptic neuron. But so you have this one, so this is just for the purpose of explaining the phenomenon. So you have one axon terminal releasing a little bit of neurotransmitter and then a little bit more, more, until you get the depolarization to reach threshold. Once you reach threshold, then the action potential is propagated. This is the temporal summation. You're adding up the effect of one axon terminal that is frequently stimulating or releasing the neurotransmitter, firing. It's frequently firing. Here, the spatial summation, you have now two axon terminals, and the two of them are firing at the same time, and therefore each of them is contributing a little bit to depolarization. So you add up the effect of these two, and they might reach threshold. So they're firing at the same area, all in one cell, correct? Exactly. So it's the effect of at one postsynaptic neuron. So if we want to represent this graphically, right? So here we have an EPSP, an excitatory postsynaptic neuron. And this one is a local potential. It goes no way. It stops because it's not strong enough. But now, if you add up 2 and 3 at the same time, frequent stimulation, you add up, you reach threshold, and then you have the spike, the depolarization to plus 30 millivolts. Now, here you have the scenario of spatial. So you have 2, 3, 4 axon terminals releasing neurotransmitter or firing at the same time, and this reaches threshold. So this is how you represent this in a graphical manner. So this happens, actually. Okay, so here you have now, this is an electron micrograph. This is beautiful. You see a postsynaptic neuron with a cell body, and each of these represents an axon terminal. So you can see that the crosstalk, or all the messages that the postsynaptic neuron receives, come from many, many different axon terminals. And here, what is shown here is the green represent excitatory EPSPs, so neurotransmitters that induce EPSPs, excitatory postsynaptic potential. The red ones are inhibitory. So you have to sum up all of these effects, the EPSPs and IPSPs. When you sum them all up, if the change in voltage reaches threshold at the axon hillock, remember here, the axon where there's a lot of voltage-gated sodium channels, so that change in polarization in voltage has to reach the axon hillock, has to reach the threshold. So it's quite complex. There's a lot of regulation, and this regulation is very, very important to prevent disease. So with epilepsy, with EPSP, with excitatory postsynaptic potential, well, if you don't generate if only one EPSP, the change in potential is not strong enough to reach threshold, then nothing happens. Exactly. And that's very important because, as I said, you don't want to have us responding to all kinds of changes that occur in our external and internal environment. We only generate a response to those changes that are strong enough to induce threshold, to reach threshold. So that's very, very important, that threshold layer parameter or plateau is very important to prevent us from responding to minor changes that might occur in the environment, external or internal. Does this make sense? Okay, so here you see another way, the postsynaptic neuron, these three axon terminals, they are excitatory. You add them up all together. So this would be spatial summation, and they will reach threshold and the action potential will travel down the axon. Here, one is inhibitory, one is IPSP. Therefore, it will counteract the effect of these two EPSPs and nothing will go through. So that's why we call the postsynaptic neuron the integrator. Okay. So the integrator sums up the effect of the presynaptic neurons. If they generate EPSP and IPSP, they are added up, the summation, and if it reaches threshold, then the nerve impulse, the action potential, will travel down the axon. Okay, so now let's describe this formation of networks of neurons, right? Think about the electricity in a house. So it comes from, let's say, from a generator, and you have a wire that brings the electricity to the dining room, or it brings the electricity to the bathroom or to your bedroom. And it's not just one wire, it goes from site A to site B to site C and so on. So the same in the brain. So you just have to think about this neuronal network as being electrical wires that are conducting these action potentials to different areas of the brain. Okay. So to clarify, temporal summation involves only one axon terminal, while spatial summation involves multiple. Yes. However, it seems all postsynaptic neurons are connected, many presynaptic neurons. So would all other presynaptic neurons not provide any impulse? No. So the sum is not one neuron. It's all the sum of all the presynaptic neurons together that are communicating, that are altering the postsynaptic neuron. It's not one neuron, one pre to one post. It can be many having this communication. And most often it's many acting together. Okay, so what is a neuronal circuit? So it's the wiring of the brain coming from the periphery. How does it reach a particular area in the brain? And so remember, it doesn't matter what kind of stimulus, what kind of, the stimulus is going to be converted into an electrical current. So how the brain can interpret is where is that electrical current that is generated by stimulus A. It could be light, it could be sound, it could be touch, it could be too much carbon dioxide. How can the brain interpret that? Because all of those stimuli are going to be converted, the effect is going to be converted into an electrical current. So it all depends on where that action potential, that electrical current, where is it going to end up? So here, what we're showing is the ear, the vestibular apparatus. So this is the hearing receptor. So here's the sensory neuron, you see, with the cell body outside. Now the action potential, when the sound is strong enough to activate the hair cells, which are the receptors for sound, then the action potential travels. And you see here, you have the axon terminal being bifurcated. Some of it goes to the cerebellum. You see this area of the brain, it's called the cerebellum. Remember, the cerebellum is dedicated to fine-tuning movement, so motor coordination. So some of this sound, this electrical current that is initiated by a sound wave, is going to be conducted to the cerebellum. Some of it is going to be conducted to the pons. This is the hindbrain, this area here, to the pons. Now, from the pons, you see that you have interneurons that are going to conduct the electrical current coming from the sensory neuron is now going to be transmitted through interneurons. Remember, interneurons are contained in the CNS, the cell body and processes. So the action potential is going to be conducted to the thalamus. This is where all the electrical current generated at the periphery, except for olfactory, for olfaction, they're all going to end up in the thalamus. And then from the thalamus, it's going to be distributed to different areas of the brain. So it all depends on this neuronal circuit. So this is something that many investigators are trying to resolve, to decipher. What are these neural circuits that conduct the electrical stimulus generated by different stimuli? So this is called the connectome. So where in the brain is like a map of sensations, right? Each area. So this is an image, a side view of the brain, and it's broken up into mosaic, into a mosaic. And each area you see here that is given a color. Of course, the brain doesn't have this color, right? So the pink area is dedicated to hearing. The green area is dedicated to touch. The blue area is dedicated to vision. So when the electrical current that is generated by light, by activation, by the photons, activating the photoreceptors at the retina in the eye, you have an electrical current that is generated, and that circuit is going to conduct the electrical current right to the occipital lobe, where is the vision. This area of the brain is dedicated to vision. Okay, so the neural code, that means where are all these action potentials going to end up? It's made up of 100 billion neurons. There's nothing in the world, in the universe that we know of that is as complex as our brain. 100 billion neurons. How do we know that the circuit goes from A to B to C to D, right? That's very hard to study. And I'm just going to show here, let's see, line 22. So you see how complex. You have 100 billion neurons, 100 billion connections going from A to B to C to D. And you see how one can map the different areas of the brain. And how can we know this? By doing functional MRIs. So for example, think of a patient that has problems with grammar, right? So let's say the word duck. To duck is to hide, right? I'm ducking. You cannot see me. I'm ducking. But, so that's a noun. But there's the duck, the animal. It's the same sound. How can the brain differentiate between a verb and a noun? So it all depends the different areas of the brain that are going to light up. And how is this determined? Is by patients or people that have different deficiencies. And then it can be the MRIs or of these patients, of these people, when we know that this area is not lighting up in response to this stimulus. And that's how you can get to a conclusion of which area of the brain is responding to different stimulus. Okay, so I hope I'm not confusing, but this is all fascinating to me. Okay, so how can these neurons, these networks, how can they be organized? So we have 5 different ways that I'm showing here. Diverging. So you see here one neuron synapsing with 2, then these 2 synapse with 3 more. So this type of neuronal pool, of this neuronal network, is an example is motor neurons. So what is the idea here is that one neuron can give rise, can control the action potential of many neurons that will affect different muscle fibers. So you have one neuron controlling different muscle fibers. Neuroplasticity, we'll come to this. Just be patient. Right, so neuroplasticity, I'm going to talk about this very, very soon. So here you have a diverging pool. Now you have converging, so you get information from many neurons that converge on one postsynaptic neuron. This is for precision, because you're getting information from many different neurons, right? Many photoreceptors are converging to give information. So this is vision, precision. Then you have a serial going from 1 to 1 to 1. This is usually sensory, an arrangement, a pool in sensory perception. Parallel for mathematics, so now we're getting really abstract. So you have one synapsing with 3, and each of these with 3 others. So this is parallel. It's different from here, from diverging. This is parallel mathematics. And this network is very interesting. So this kind of a pool of network is involved in generating memory. Why? Because you have one neuron synapsing with this one. And this one has a collateral. So it will synapse with this, but then the action potential goes back to the first one, and it continues going back and forth to generate a very strong synapse. And that's what is necessary to generate memory. Okay, here we go, synaptic plasticity. So these connections among neurons are not static, they're very dynamic. So during the day, we have all kinds of experiences, and we will not remember all of them tomorrow or in a month or in a week. Why? Because these connections, these synapses change. If they are weak, if they're not strong enough, they will be destroyed by microglia. If they are physically strong enough, then they will form long-term memory. That's what's known as synaptic plasticity. That means the formation of synapse can vary. So let's look. I thought this was a very interesting short video that shows the concept of synaptic plasticity. So let me show you. So here you have a neuron with the cell body and the axon. And let's say you're studying names of muscles, and you have to reverberate. You have to use that circuitry to remember memory. Go back and forth, back and back and back, memorization. So what happens, you see here the formation of synapses, and then they break apart, and then they make synapses with other dendrites, with other neurons. And this is constantly going on in our brain. The synapses are forming and breaking until they are very strong, and then they will be permanent. Okay, so this brings us to short and long-term memory. So short-term memory resides in this area of the brain that's known as the hippocampus, because it looks like, you don't get idea here, it looks like a seahorse. It depends on the angle that you're looking at. So the hippocampus and the amygdala. Amygdala is very much dedicated to emotion. Okay, so the green part is the hippocampus, the red is amygdala, and this is where short-term memory resides. So that means things that happen and we remember, but then we forget after tomorrow or after 10 minutes or whatever. But solid memory, long-term memory, involves the prefrontal cortex. And so how can this happen? The concept of short-term memory and long-term memory. So this was identified or discovered by Eric Kandel, who's a professor at Columbia. He won the Nobel Prize for this. So instead of asking the question in a human brain, how is memory long-term versus short-term memory established, because that would be possible because we have billions of neurons, he studied on this snail called Aplysia that had a very simple network of neurons. So what he was able to demonstrate is that short-term memory that resides in the hippocampus and the amygdala, that means that the change in synapses is just electrical and chemical. There's just a change in chemical, the neurotransmitters that are released, and the action potential. So it's not permanent. But the long-term memory, you have an anatomical, a change. You see here the axon, the neuron, the presynaptic, and you see that the synapse increases because you constantly sending stimulus. So you constantly repeating and repeating in such a way that the actual shape of the synapse changes. And so that's what leads to long-term memory. And you see here, this is a beautiful image of a dendrite. And you see here the synapses, the shape changes. So when the shape increases, you actually see the change. It's a mushroom-like of head. That means it's a permanent synapse. So the shape of the synapses of the presynaptic neurons, the shape here changes with the strength of the signal. Can you forget something in long-term memory? Well, in Alzheimer's, what happens is you lose short-term memory. So that many patients can recognize the family, but they cannot recognize someone that they saw yesterday, for example. So they lose short-term memory. But as the process degenerates, then the long-term memory will go. So some patients with Alzheimer's can remember, of course, all this depends on the level, right? They can remember songs that they sang when they were children, but they won't remember something that happened yesterday. So the first thing to go is the short-term memory. The long-term memory resides for longer because of the strength of the synapses that are made. Okay, so this is what is represented here. This is the hippocampus and the amygdala. This is another area, which is the entorhinal cortex. It is also associated with memory. So here you see, this is a PET scan. Remember positron emission tomography, where you inject a patient with a radioactive form of glucose. And because the brain is so active, the cells will take up this radioactive glucose. If they are more active, you have a greater signal that is represented by the red color. If the brain is not so active, if the neurons are not so active, they will not take so much glucose. So this is a normal brain. Remember, in the brain, the gray matter is on the outside. And the red area is where there's a lot of activity going on, a lot of action potentials. So taking up a lot of glucose. Now, this is the brain of an Alzheimer's patient. Only a few areas remain active. So it's a big, big difference. And we don't know why this happens. So this is the brain of a normal patient. And here you can see the shrinking of the brain, the size of a patient with Alzheimer's. And we are only absolutely sure that the patient has Alzheimer's post-mortem, although nowadays there's many different tests that can be used. But it seems to be due to the inability of clearing the brain from debris. So you see the appearance of these plaques that are known as senile plaques outside the brain, and these are neurofibrillary tangles, which are aggregates of proteins inside the neurons. So these are, let's say, the hallmarks of Alzheimer's disease. And I would highly suggest that you look at this video that really explains Alzheimer's. Okay, so as we age, I wanted to get here. Yes, let me finish these slides. So as we age, we lose a lot of capacities. Our neurons start to degenerate. The reflex time, because the propagation of the action potential is not so efficient. So the reflex time increases. And also the organs, the receptors also deteriorate. And in humans, it seems that there is some hope that we have some stem cells, some precursors, especially at specific sites, for example, the hippocampus, that is involved in short-term memory, near the olfactory bulb, and also in the basal ganglia. It seems that there's some stem cells that, depending on different stimuli, they can be differentiated into oligodendrocytes, astrocytes, neurons. So this is being studied greatly because it's very important to see, can we activate these stem cells to generate the neurons that have been disappearing. And you see what happens, the process of neurogenesis, the formation of new neurons. This is very complicated. Okay, so you might have new neurons being formed, but then they have to reach the specific site. And not only that, they have to make connections with the specific neurons. And during development, there's cues that are generated that allow for the neurons, the axons, the processes to grow in specific directions. And when we are an adult and we put in new neurons, those cues might not be available already. Okay, so one important thing to keep our neurons working and maybe to have these stem cells, these precursors being active, is to have an enriched environment. So this is an experiment that was done with mice, where some mice were put in a cage with no toys. And this is the hippocampus, a section, and a histological section of the hippocampus. The green shows neurons, the red shows new neurons. So they are differentiated from those stem cells. So we know that in rodents, for sure, they are stem cells at the hippocampus. They can be differentiated into neurons. So if you have a rich environment, like in this cage where the mice have all kinds of toys, then they are active. So activation will induce the differentiation of new neurons. But if the environment is quiet and there's no activity, it's the same activity, it's the same idea of muscles. If you don't use them, you lose them. So having an enriched environment seems to be very important to the maintenance and to maybe the awakening of these stem cells. So I think we'll stop here and we'll have the spinal cord for Thursday. Okay, any questions? Okay. Okay, see you Thursday. Bye-bye.